Hello everyone, this is your educator Priyanka Thakur and I'm teaching you life sciences and welcome you all on our YouTube channel Gurmantra Shiksha Ka Granth. So today in this video we are going to cover auxin, its biosynthesis, transport and physiological effect. Okay, so I hope you all will enjoy this, you all are going to enjoy this video. And before starting the video, let me tell you what we have covered till now. So this is the section 4 plant physiology and biochemistry of botany syllabus, and this is section 456 of zoology syllabus. Okay, we have covered 4, 5, 6, all these three sections. Me and Saranya ma'am both have covered these three sections, and covering section 4 of botany syllabus, we have covered C3 C4 cycle, photorespiration, respiration, glycolysis, TCA cycle. We have covered all these things, and I'm going to start biosynthesis, mechanism of action and physiological effects of auxin. In this video we will talk about its biosynthesis, physiological effects, and uh, about its mechanism of action we will discuss in some other video. Okay, so let's start with our today's topic, which is auxin, our phytohormones. Okay, what exactly are phytohormones? These phytohormones, these are actually organic substances which are synthesized by some specific plant or plant cells or tissue, and these substances are active in a very low concentration. They can promote or inhibit the development of a plant. Okay, so these phytohormone can be stimulatory or inhibitory. We cannot say that will only promote the function of any particular organ or tissue. They can even inhibit some developmental process of plant. So how these hormones were discovered? So Charles Darwin and Francis Darwin in 1880, they were doing some experiments on phototropism, and their experiments on phototropism led to the discovery of phytohormones. They were actually performing their experiments on the colutile of canary grass, Phalaris canariensis. This is the shoot and this is the colutile, and you know that the protective covering or shoot is known as colutile. So they actually studied the banding of canary grass colutile. As I told you, they were studying the phototropism. So they saw that the colutile of canary grass was banding towards a unilateral source of light. Okay, so it was banding toward the unilateral source of the light. Fine. After that, number of scientists performed different experiments, and finally the F.W. Went uh, discovered the very first plant hormone in the oats colutile, Avena sativa, and the hormone which he discovered was auxin. Clear? Auxin was the first hormone which was discovered, and it was discovered by F. Went in the colutile of oats. So don't get confused with the canary grass and oats. So canary grass was the first plant on which experiments were conducted, and with those experiments led to the discovery of our phytohormones. But exactly, phytohormones were not discovered in the canary grass. It was oat seedling, oat colutile, in which phytohormones were discovered. And who discovered? F. Went was the person who discovered and isolated our very first hormone, which is auxin. Clear? So auxin, what is the meaning of auxin? Auxin means to increase. Why it was named so? Because the chemical which was isolated by Went in oat was actually promoting the elongation of colutile, because it promotes the elongation of colutile. It causes an increase in the colutile. That's why it was called auxin. Natural occurring auxins are indole acetic acid, indole butyric acid, and phenyl acetic acid. The most abundant auxin that is found in the cell is indole acetic acid, and auxin is mainly synthesized in shoot, apical meristem, young leaves, fruits, and seeds. So remember, auxin is synthesized in shoot, apical meristem, young leaves, fruits, and seed. It can be found in both chloroplast and cytosol. Get it? In cytosol, you will see auxin in a conjugated form. It means that auxin can occur in a conjugated form as well as in the free form. This is a diagram showing that unilateral of source, how it can cause the bending in the colloidal. It will be concentrated toward one region, and in that region it will cause the elongation of the cell, which ultimately results in the elongation of colloidal. Okay, so let's talk about biosynthesis of auxin. We have two pathways. It's not dependent, it's independent here. There are two pathways of biosynthesis of auxin. One is tryptophan dependent pathway, other one is tryptophan independent pathway. As we are not so much sure about independent pathway, like we do not have a lot of information about tryptophan independent pathway of auxin biosynthesis, so we will mainly discuss about tryptophan dependent pathway in this video. So tryptophan dependent pathway includes indole 3 pyruvate pathway, indole 3 pyruvic acid pathway, you can say. Second one is tryptamine pathway. Third one is indole 3 acetonitrile pathway. And fourth one is indole 3 acetamide pathway. Is it clear to you? Okay, fine. Indole 3 pyruvate pathway. All these pathways are named on the basis of their intermediate products. Okay. So let's start our tryptophan dependent pathway. But before starting any of the pathway, let me tell you, either it is a tryptophan dependent or it is a tryptophan independent pathway. Here there should be independent and not dependent. Both the pathways will start from chorismate. Okay. First one will be the chorismate, okay, the first molecule which will ultimately lead to the formation of tryptophan, and then tryptophan will lead to the formation of indole acetic acid, okay. In tryptophan independent pathway, there is no role of tryptophan, but in tryptophan dependent pathway, we will see that in each and every pathway, tryptophan will be ultimately leading to the formation of indole acetic acid, okay. So chorismate will be converted to anthranilate, okay. Anthranilate in the presence of enzyme anthranilate synthase, okay. Chorismate will be converted to anthranilate in the presence of enzyme anthranilate synthase, and then anthranilate phosphoribosyl phosphoribosyl transferase will act on anthranilate, and it will result in the formation of 5-phosphoribosyl anthranilate, and on this phosphoribosyl anthranilate isomerase can act, and it will then be converted into 1-O-carboxyphenyl amino 1-deoxyribulose 5-phosphate, okay. Now this indole 3 glycerol phosphate synthase, it can act on 5-phosphoribosyl anthranilate or on 1-O-carboxyphenyl amino 1-deoxyribulose 5-phosphate, okay. It can act on both, and they will result in the formation of indole 3 glycerol phosphate, okay. When this indole 3 glycerol phosphate combined with serine, it results in the formation of tryptophan. So do remember this pathway, how tryptophan is formed. This is a tryptophan synthesis in plastid, okay. So chorismate will then be converted into anthranilate, which is converted into 5-phosphoribosyl anthranilate, and which is isomerized into 1-O-carboxyphenyl amino 1-deoxyribulose 5 phosphate, which in the presence of indole 3 glycerol phosphate will be forming indole 3 glycerol phosphate, and along with serine, indole 3 glycerol phosphate will result in the formation of tryptophan. Fine, we have our tryptophan now. Now we can discuss about the tryptophan dependent synthesis of auxin. So this is our very first pathway, indole 3 pyruvate pathway. Here The tryptophan undergoes a deamination reaction in the presence of tryptophan transaminase enzyme, and after the deamination of tryptophan there is the formation of indole 3 pyruvate, which will undergo decarboxylation reaction in the presence of enzyme decarboxylase, and indole 3 acetaldehyde is now formed. Indole 3 acetaldehyde will be dehydrogenized by indole acetic acid dehydrogenase, and then indole 3 acetaldehyde will be converted into indole 3 acetic acid. Okay, indole 3 acetic acid. Is it clear? Tryptophan will undergo deamination and indole 3 pyruvate will be formed, which will undergo decarboxylation, and then indole 3 acetaldehyde will form, which is dehydrogenized by indole 3 acetic acid dehydrogenase, and indole 3 acetic acid is formed. Clear? Further, in indole 3 pyruvate pathway, you will see deamination, decarboxylation, and dehydrogenation. Next pathway is tryptamine pathway. What happens during tryptamine pathway? Tryptophan. In indole 3 pyruvic acid, we saw that second step was decarboxylation and first step was deamination. But here in the tryptamine pathway, first step is of decarboxylation and then occurs deamination. So very first tryptophan will be decarboxylated into tryptamine, and that tryptamine will be deaminated into indole 3 acetaldehyde. Fine. And then indole 3 acetaldehyde will be dehydrogenized into indole 3 acetic acid. So do remember what happens during indole 3 pyruvic acid pathway and tryptamine pathway. In the indole 3 pyruvic pathway, first step is deamination. In tryptamine, it's decarboxylation. But in IPA pathway, second step is decarboxylation, while in tryptamine pathway, it's deamination. Third step is common to both in IPA pathway as well as tryptamine. It's dehydrogenation of indole 3 acetaldehyde, which will be converted into indole 3 acetic acid. Okay. Next is our indole 3 acetonitrile pathway. So in this tryptophan in the presence of some enzyme will convert into indole 3 acetonitrile. So the name of all the pathway has been given on the basis of their intermediate product. Okay, so indole 3 acetonitrile in the presence of nitrulase enzyme will be converted into indole 3 acetic acid, and this pathway, indole 3 acetonitrile pathway, is generally seen in the mustard family, Brassicaceae family. This can be asked to you in any of the examinations, so do remember this thing, that indole 3 acetonitrile pathway is commonly seen in the mustard family. Okay, so in this tryptophan is converted into indole 3 acetonitrile, which is in presence of nitrulase enzyme is converted to indole 3 acetic acid. I hope you're not having any confusion now. This is our last pathway. This is the fourth one, actually, not the third one. I should have written it's the fourth one. IAM pathway, indole 3 acetamide pathway. In this, tryptophan in the presence of monooxygenase get converted to indole 3 acetamide. Fine. Tryptophan in the presence of an enzyme monooxygenase get converted to indole 3 acetamide, which is then converted into indole 3 acetic acid. This pathway you can see in the agrobacterium. Okay, so I hope all the four pathways are now clear to you, and as I have already told you that we are not going to discuss the tryptophan independent pathway because we have not enough information about the tryptophan independent pathway, but we discussed all the four pathways of auxin biosynthesis which are dependent on the tryptophan. Along with this, we also got to know how tryptophan is formed. All the pathways will start from the chorismate. Okay, chorismate will ultimately lead to the formation of tryptophan. Okay, fine. So this is all about biosynthesis of auxin, and next we are going to talk about its transport. Okay, so auxin transport in plant is predominantly polar. Okay, what polar means, it occurs in cell to cell fashion. Okay, the transport of auxin in plant is predominantly polar, which occurs in a cell to cell fashion. In a stem, the polar transport in stem, this is the stem, okay, stem portion, the polar transport will be basipetal, while in the root it will be acropetal. And now what is basipetal and acropetal? See, this is the base for both root and shoot. In case of a basipetal transport, basipetal polar transport, which usually we see in the shoot, So here, the transport of oxygen takes place from the apex to the base. Okay, this is the basipetal transport. And in the acropetal polar transport, the transport occurs from base to the apex. So this is the base for both root and shoot, as I already told you. Okay, so now in shoot, the transport of oxygen will be polar, unidirectional, but from base to apex. So from the transport from base to apex we call it acropetal, and the transport from apex to base we call it basipetal. So in shoot we will see polar basipetal transport, while in a root we will see a polar acropetal transport. Okay, fine. Polar transport occurs through vascular parenchyma. Vascular bundle is represented by xylem and phloem. So definitely xylem and phloem. Parenchyma will be responsible for the polar transport of oxygen. And in shoot, the tissue which is responsible for polar transport is xylem parenchyma, and the in shoot the tissue is phloem parenchyma. Remember, polar transport is energy dependent. It means it is an active process. Polar transport is an active process. It is energy dependent, but it is independent of gravity. Remember this thing clearly, very clearly, that polar transport is energy dependent, but it is independent of gravity. Do not forget this thing. Okay, in many of books you read that oxygen can be transported in a non-polar manner. Okay, you read about non-polar transport of oxygen, but this case, the non-polar transport of oxygen, whenever you are given with a given a MCQ and there is no such option both polar and non-polar, mostly you tick on the polar transport. Why? Because you have read mainly about the polar transport. There are not enough evidences of non-polar transport of the oxygen, but still it is rarely seen, it was shown by Goldsmith and his co-workers in 1974 in Coleus species, and he proved the non-polar transport of the oxygen. But after that, then nobody else reported such kind of transport of the oxygen. But still, he had proven, he along with his workers had proven the non-polar transport of the oxygen in Coleus species. And do remember one thing, that non-polar transport is it occurs mainly via phloem. Okay, it is not xylem based. Polar transport is both xylem and phloem based, but non-polar transport is mainly phloem based. Okay, and one other thing that you have to remember is that polar transport is much slower as compared to the non-polar transport. Non-polar transport of oxygen is very fast compared to the polar transport. So do remember this thing, that polar transport is slower than the non-polar transport, which is too fast as compared to the polar transport. And as we know that polar transport is energy dependent, on the other hand non-polar transport is energy independent, and it is known by another name, long distance source to sink transport. And you know that from source to sink there is usually the function of phloem. That's why we call that non-polar, we say that non-polar transport is phloem based. It is also known as long distance source to sink transport. If they are asking you that long distance source to sink transport is a polar one or non-polar one, then remember you have to answer this is a non-polar transport. Clear? Okay, I hope the basics of the transport are clear to you now. And this oxygen transport can be demonstrated by a chemiosmotic model. Okay, so what it actually tells us, this chemiosmotic model, it says that oxygen influx is driven by proton motive forces. By what? Proton motive forces. Fine. Now, what are these proton motive forces? It means oxygen influx is dependent upon membrane potential differences as well as the pH. Okay, and efflux is driven by only differences in membrane potential. There is no role of pH in the efflux, but pH does play a role in the influx of the oxygen. And along with the membrane potential, efflux also depends upon some specific auxin efflux proteins which will be present at the base of the conducting cells. Conducting cells are going to be xylem or phloem parenchyma, and the auxin efflux protein will be present on the base and not on the apex of the cells. I hope you are understanding what exactly I mean by base of conducting cells. If you're not getting it, you will see it in the image, so you will be easily understanding what I'm trying to tell you. Okay, so auxin influx is dependent upon mainly by proton motive forces, which will be including both the membrane potential and pH, while the efflux is mainly driven by the membrane potential and pH doesn't play any role in the efflux. Along with that, auxin efflux proteins will be doing a great role in the efflux of the auxin. So auxin influx occurs in two forms. It will be in protonated form or deprotonated form. So protonated form is actually lipophilic, and lipophilic forms can enter the cell by a simple diffusion. It means we do not require any energy for the transport of protonated form, but deprotonated form do require some source, some proteins, because they cannot enter the cell by themselves. They can cross the membrane themselves. They enter through secondary active transporters, co-transporter. Okay, so deprotonated form of indole acetic acid will enter the cell through secondary active transport via protoindole acetic acid symporter 2H positive IAA symporter. For example, OX1. OX1 is one of the proton indole acetic acid symporters. Okay, remember they will be the symport. You know, symport when the transfer of molecules occur in the same direction. Okay, but one will be traveling from lower concentration to higher concentration, and other will be traveling along the concentration gradient. Okay, so deprotonated form will enter they will enter through secondary active transport. Okay, they will enter into the cell by a secondary active transport using this proton indole acetic acid symporter. Okay, for example, OX1. So auxin and this is about auxin influx, how influx will occur. Now let's talk about auxin efflux. It is mediated by pin proteins and PGP. Pin proteins are actually integral membrane proteins which are distributed on the basal end of the cells. As I told you that auxin efflux proteins will be responsible for the efflux of the auxin which will be present at the base of the conducting cells. So these are pin proteins which are integral membrane protein and are distributed on the basal ends of the cell. So we call we say that the distribution of pin protein is polar distribution. Okay. We can see 8 types of pin proteins in Arabidopsis, and they start from pin 1 to pin 8. Okay, PGP proteins, P glycoproteins, they are also known as ABC, ABCB, ATP binding cassettes subfamily B proteins, and these are distributed uniformly on all the end of the cell, so their distribution is non-polar. We cannot say that their distribution is polar. So now you got to know how auxin is going to be going to enter in the cell and how it will be exiting the cell. So you know about our proton indole acetic acid symporter. So let's talk about it further. Okay, this is the chemiosmotic model which I have taken from the Theis and Zyger plant physiology book. I hope I'll be able to make everything clear to you. It's fine. This is the cell wall. This is the cytosol. Okay, this one is the vacuole here. Fine. So let's talk about it. As you can see, deep protonated form of indole acetic acid is higher in the cytosol. Okay, it is higher in the cytosol. So indole acetic acid can enter the cell passively. It can enter the cell passively. We know that protonated form is lipophilic and it can enter the cell passively, while deprotonated form require symporters. What does it require? It requires symporter. This is the symporter in which protons will enter from their lower concentration to higher concentration. Okay, protons will be entering from their lower concentration to higher concentration. So this is a secondary active transport. Permease hydrogen co-transport. Okay, so indole acetic acid will be entering through this importer into the cell. So, indole acetic acid is mainly found in deprotonated form in the cytosol, but it is found in protonated form in the cell wall. In cell wall, pH is 5, while in cytosol is pH is 7. Why it is in deprotonated form in the cytosol, and why the pH is 5 in cell wall and 7 in cytosol? This is because on the membrane there are some proton pumps, okay, ATPase, H positive ATPase, okay, which will be pumping out the hydrogen ion from the cytosol to out of the cell. So in cell wall the pH will be 5, while in the cell cytosol pH will be 7, which is a neutral pH, and the indole acetic acid is mainly found in the deprotonated form, while outside of the cell in the cell wall it is found in the protonated form, because there is enough hydrogen ions, while hydrogen ions are all removed through the H positive ATPases out of the cytosol. Clear? This is the protein. These are the proteins which are present on the base. These are pin proteins which are present on the base of the cell and not on the apex, which will be resulting in the efflux of the auxins. Now I hope everything is clear to you now, where our pin proteins are present, and where protonated form will be present, where in a deprotonated form will be present, what will be the pH of the cell wall, what will be the pH of our cytosol, not our cytosol but cell cytosol, and why the pH is 7 inside the cytosol, why there are more deprotonated form. I hope all these things are clear to you now. But the one other important thing, that chemiosmotic model is one important thing is that this transport can be inhibited by some inhibitors, which are TIBA and NPA. These are synthetic compounds and not found naturally in the cell. TIBA means 2,3-triiodobenzoic acid. 2,3,5 triiodobenzoic acid, and NPA means naphthyl thalamic acid. Okay, naphthyl thalamic acid. 2,3,5 triiodobenzoic acid and naphthalamic acid. These are the inhibitors of the polar transport of the auxin, but are not naturally found in the cell. These are synthetic inhibitors. Clear? So that is all about the transport of the auxin. Let's talk about the effects of auxin now. So auxin, the very first and important is cell elongation. Auxin results in the cell elongation, that's why it has been named so. And how it causes the cell elongation? At lower pH, the expansins proteins found in the cell get activated. Okay, these proteins results in what? They will result in the they will disrupt the hydrogen bonding between the cellulose microfibril, and causing the laminate structure of the cell wall to loosen. Okay, so with the rigidity of the wall reduced, the cell can elongate now. Fine. So next effect of auxin is it can induce differentiation of xylem in high concentration, while it induces differentiation of phloem in low concentration. Remember this point. Higher the auxin, it will be differentiating xylem. Lower, in low concentration, it will be differentiating phloem. Okay, auxin can stimulate the root initiation in the stem cuttings and lateral root formation in tissue culture, but they inhibit primary root elongation. They inhibit primary root elongation, stimulate lateral root formation, because as we know that lateral roots are formed from the pericycle, so they stimulate, auxin molecules stimulate the pericycle to form the lateral roots. Okay, next is apical dominance. The very important physiological effect of the auxin is apical dominance. What happens in apical dominance? The apical bud keeps on increasing, elongating, while the lateral buds are inhibited. Lateral bud growth is inhibited. Okay, now how this auxin inhibit the lateral bud growth? Actually, it stimulates strigolactones, and these strigolactones activate the branched 1 genes, BRC1 gene, and the product of these BRC1 genes inhibit the lateral bud growth. Clear? Apical dominance, how auxin can cause apical dominance? When there is abundant auxin, it will be stimulating the strigolactone will be activating the branched 1 genes, whose product will be inhibiting the lateral bud growth. So you have seen gardeners doing pruning. That pruning, you have seen in tea leaves we require pruning. Why? Because it will result in the lateral bud growth. Apical bud will be removed, and hence auxin formation will be reduced. That's how the strigolactones will be reduced, and branch chain will not be able to perform its function of inhibiting the lateral bud growth. And now there will be a resume in the growth of lateral buds. So in bushing, or if we want more and more lateral leaves, there will be removal of the tip of the plant. Okay, so it promote the development of fruit set, and it can also induce parthenocopy. Auxin induces a tropic response. Okay, not triple response. Keep in your mind, it's not triple response. You can attempt a question wrongly because you think it's triple response. It's not triple response, it's a tropic response. Fine. What happens during tropic response? In tropic response means there will be the differential growth in plant organs under the influence of directional stimulus such as light and gravity. Clear? Tropic response, as we have seen that Darwins were experimenting on phototropism while they were performing their experiments on the canary grass. Okay, so that was the tropic response of the auxin, in which there is a differential growth in the plant organs under the influence of some directional stimulus such as light and gravity. Along with this, auxin can promote the ethylene production. When auxin is present in a higher concentration, when its on concentration increases, there will be the production of ethylene now. And one other important function is it can promote the femaleness in the dioecious flowers by ethylene. Okay, I hope and everything is clear to you now about the biosynthesis of auxin, about how the transport of auxin will takes place, and about the physiological effects of the auxin. This is all for today. We will be discussing the signaling pathway of auxin in some other video. Okay, so before going, let me tell you that Neha Tanisha ma'am is taking her classes on Unacademy, and you can see the detailed paid classes information here. Along with this, I hope you have seen that there is alarming notification regarding the hike of the fee for CSIR net coaching. And you can see there is a good timing, watch time of Nihat Danesha ma'am. She is a too good educator, as you all know. If you don't know, then I'm telling you, do watch her videos. You will get to know yourself this thing. If you want a discount, you can use the referral code NEHA, so that you will be able to get 10% off. Okay, along with this, to subscribe to our channel, do not forget to press the bell icon to get the notification of the videos. Along with this, you can check the description box and can join us wherever you want. Thank you. Till then, take care. Bye bye.